Hello, everyone. This video is for January 13th or 14th. What we did today was start off with some announcements. Then I went through and just reviewed Punnett squares. So our notes from the previous class, we talked about Punnett squares, how to do them, how to read them. So I want to go over that just a little bit before I give you time to work on Punnett practice. I had originally had another plan for today, but the technology was not wanting to partake in that. So this was all we did. I gave you plenty of time to work on this. So the only announcement I have today was that we don't have school on Monday. We have a long weekend. So don't come to school on Monday. See you Tuesday, though. In this review, we're going to create a Punnett square crossing these two parents, just meaning that these are the two parents that are going to combine and make babies. And we're going to work through and figure out the phenotypic and genotypic percentages and ratios that we see in this cross, this Punnett square. And the phenotype that we're working with today are dimples. So the dominant allele is going to mean that we have dimples. The recessive allele is no dimples. So dominant trait, we do have dimples. Recessive trait, we do not have dimples. Before we move on, I want to get some extra practices with how to differentiate between genotypes and phenotypes. So we're going to start with all the possible genotypes. Remember, genotypes, geno means gene. So this is going to be our gene makeup. And what our genotypes look like come from our alleles. So remember that these two versions of a gene are called alleles. We have a dominant and we have a recessive version. Now, how do we get the different possible genotypes? Essentially, what we're looking at here is in what different combinations can these two alleles come together to give us different, like I said, combinations. For example, we can get a big B from our mom and a little b from our dad and have this genotype. Very similarly, we get a big B from our mom and also a big B from our dad and have that genotype. We can also get a small b from our mom, small b from our dad to get that one. So these are the different combinations of genotypes. Now, because we don't always use the same letters when it comes to genotypes, we can't just call them big B, big B, big B, little b, little b, little b, because that's a mouthful and people are not always going to know what you're talking about. So we refer to these different genotypes using specific vocab words. This one we call homozygous dominant. It's homo because homo means the same. And you can see that they're both the same allele. They're both big B's. So not only are they homozygous, they're also dominant because they're both dominant alleles. So that one we would call homozygous dominant. Homo because they're the same, and dominant to specify that they're both dominant alleles instead of recessive alleles. Very similarly, if we look at this genotype, it's also going to be homozygous. Again, because it's the same. But this one, because we have two little b's or two recessive alleles, we call this one recessive. Homozygous recessive. All right. For the last one, the middle one here, you may notice that these two alleles are not the same. They are different. So that's why we call these heterozygous. Because hetero means different. Now for these, we don't have to specify whether it's dominant or recessive, because if you use the term heterozygous, you automatically know that you have one dominant allele and one recessive allele. Okay, and that is how you figure out the possible genotypes. So I'm going to do that. Bam. So there are genotypes. Then, what are all the possible phenotypes? Phenotypes are our physical characteristics. Okay, so up here it tells us what physical characteristic we're looking at here. So our phenotypes will be either we have dimples or we don't have dimples. Those are our only options in this problem. Either have them or you don't. There's no in between. Okay, so that's how you know genotypes and phenotypes. So let's go ahead and jump to our Punnett square. So I have included the steps here, so we just are doing everything on this one slide. So we are crossing a homozygous dominant parent with a heterozygous parent. So those are the genotypes. Okay, so I'm going to make a note here. These are the genotypes. Okay. To put a little bit more information on this, we can also figure out these parents, do they have dimples or don't they? So keep in mind that having dimples is a dominant trait. So every time we see this dominant allele, we know that that organism is going to have dimples. So if we're looking at this homozygous dominant parent, we have at least one dominant allele, so we know this person definitely has dimples. I'm going to write that better. Hold on. Bam. Now this heterozygous is where a lot of people get confused because they see it and they think, well, we have one dominant allele, but also one recessive allele. So what is it? And again, just remember that every time we're talking about dominant alleles, every time you see a dominant allele, that dominant trait is going to show. It's almost as if this one doesn't exist. Almost. It still does, but we don't see it. So this parent is also going to have dimples. So in this Punnett square, we are looking at two parents, both of which have dimples, and we're trying to figure out what is the possibility that their children will have dimples, and is there a possibility that their children might not have dimples? That's what we're answering with this Punnett square. So I'm going to clear this up. Bam. So step one is going to be to set up your Punnett square. So if you do not have this box set up already, you want to go ahead and make sure to do one. For the most part, I'll give you one, but in case you're not given one, don't forget the Punnett square. Now we're going to put these parents on those Punnett squares. And it doesn't matter where you put which parent, but one will go up at the top and one will go on the left-hand side here. So you're going to bring the alleles from the top genotype down. You're just going to drag them down. So this big B will be in the first box and the second box. Same with this big B over here. I'm going to drag those down. For the genotype on the left, same idea, except you are dragging them to the side. So we're going to take this big B over to this first box and again to the second. And for this little B, we're going to do the same. Take it over to this box and bring it all the way over. And when I take my arrows away, this is what your Punnett square looks like. So now we're going to calculate and write out our percentages and ratios. So when we do that, I like to start with genotypes. So remember, genotypes are the combinations of alleles, how they come together. So in this Punnett square, so just looking at the babies that these two parents created, I only see two different genotypes. I see the homozygous dominant here, and I see the heterozygous. I don't see any of these homozygous recessive babies, so I'm not going to include those here. For the percentages, remember that each box is worth 25%, and it's kind of like 4 quarters equal $1. So here we're looking at what percentage are homozygous dominant. So you have one here and one here. All of these are definitely not, or the rest of those are definitely not homozygous dominant. So that's going to be 2 out of our 4 babies are going to be homozygous dominant. This in a percentage will be 50%. We'll do the same thing with the heterozygous. So how many are heterozygous? So here we have 1, here we have 2. So again, 2 out of 4 babies are heterozygous. That means 50% of our babies are heterozygous. For the ratio, we are looking at how many individuals are there of each. So we're going to look at all of the different possible genotypes. So we have homozygous dominant, heterozygous, and homozygous recessive. So we'll just look at our Punnett square and literally just write down the number of different genotypes. So for homozygous dominant, we have 2 individuals that are homozygous dominant. So we'll put 2. For heterozygous, we have 1 individual that's heterozygous and 2. So we have 2. And for homozygous recessive, we have none. So we're going to put 0. So our ratio for genotypes, we have 2, 2, 0. Or if you want to simplify, you could just do 1, 1, 0. And that's it for how to figure out the percentages and ratios of genotypes. Lastly are the phenotype percentages and ratios. So remember up here, these are your different options. Okay, so you either have dimples or you don't have dimples. That's our only option. So when looking at our Punnett square here, I notice that every single individual has at least 1 dominant allele. This tells me that I see babies with dimples. This also tells me that I don't see a single baby that doesn't have dimples. I don't see any homozygous recessive. So all I see are dimples. So then for our percentage, we just look at how many have dimples. So 4 out of the 4 have dimples, which makes it 100%. For the ratio, you're going to do very similar as what you did in the genotype, but instead of the genotype, we're going to do phenotype. Here, there's only two options, the dominant and the recessive, so whether you have dimples or not. And like we just said here, 4 of our babies have dimples and 0 do not. So our ratio there is 4:0. So this seems like a lot of work, but it tells us a lot of information. So we can look at what we did here and say, if we take 2 parents that have dimples, one of which is heterozygous, there is a 100% chance that their babies will have dimples. Yeah. After we reviewed the notes, like I said, I gave you plenty of time to finish that punnet practice. I showed it to you in the last video, but just in case, you can find this under modules. It's going to be under week 2, and it's called punnet practice. So for this, you'll have to use Kami. So you click on this twice to download. Make sure that something pops up in the corner of your Chromebook saying it's downloading. And then you're going to upload it into Kami, and here are the instructions there. Okay. If you need help with this, of course, let me know. Just to point out a few things, because as I was grading those that are already turned in, I noticed that some people were missing things. So for this assignment, here's my reminder to you. This information is hecka important. You need to know what the phenotypes are, like green or yellow, and you have to know which one is dominant and which one is recessive. So it tells you here. For number 1, you're going to write down all the possible genotypes, kind of like what we did earlier. Don't forget to also write down what their phenotypes are. So if you have a homozygous dominant genotype, is that going to give you a green or a yellow pea pod? Don't forget that part. Yeah. Another thing I noticed is in number 2, couple things, kind of like math, writing down your work, showing your work is going to help you out, especially if you make small mistakes. I mean, we're only human. It happens sometimes. So make sure to write your parents here so that it's a lot easier to figure out what genotypes go in these boxes. The other thing I wanted to remind you all was don't forget to write the phenotype and genotype percentages and ratios. So you're going to write down for genotype, every genotype that you see, and then the percent that you see them. So do you have 25% of one, 50% of another? Kind of like what we just did in our example. And same with the phenotype. In this case, we have greens and yellow pea pods. So write down green and yellow. Tell me what percent are green and 1% are yellow. Okay, so don't forget those things. If you have any questions or need any additional help with getting this done, don't hesitate in letting me know.